Hello, everyone. Welcome to my video tutorials for spatial dataset analysis. Today's video tutorial is for Python users because I'm going to show you how to analyze the spatial dataset using the Squidpy package. In today's video tutorial, I'm going to show you how to analyze the Nanostring CosMx dataset for the non-small cell non-cancer dataset. So first we can load the package that is required for today's analysis. We are loading NumPy pandas. Both packages are required for data manipulating. And we need the ScanPy package to analyze the single cell gene expression data. And the Squidpy is the package to analyze the spatial data. And also we import the matplotlib and the Spatial packages. Both packages are required for plotting the images. So if you want to use the dataset for practice, you can download the dataset from Nanostring website. If you go to Nanostring website, you can see they provided the non-small cell, non-cancer dataset as one of the public available datasets to demonstrate their technology. So the online tutorial uses the non-5 repeater tool. If you want to use the non-5 repeater tool, you can click here to download the data. I'm going to use the non-5 repeater 1 dataset. If you want to use this dataset, you can click this link and download the dataset. So I downloaded the data already. We can have a look at the dataset. You can see in my folder, this is the non-5 repeater 1 dataset. You can see we have 4 folders in this folder because they use the 4 antibodies and perform the immunohistochemistry to help to identify the cell boundaries. Then in this folder are the immunohistochemistry staining for 4 antibodies. We can click and have a look at the images. You can see the best staining is for the green channel. You can see this is the staining for the cytokeratin protein. Then in the other folder, we have the cell labeling, and in this folder, we have the cell overlay. We can have a look at this folder as well. You can see here we have all the images for the cell segmentation on different field of view images. You can see here in total we have 30 images. If you want to download this dataset for practice, the original dataset have 32 images, but in the expression matrix data, it only has the data for 30 images, so you need to delete the 31 and the 32 images in those folders. Otherwise, you will not be able to load the data into Python for analysis. So you can see we have the CSV file for the expression matrix data, CSV file for the field of view positions, and the metadata. So now we can go to the lab and perform the analysis. So first we can use the read nanostring function to read the data and create a data object. You can see the path will be the files directory to show where we saved the data we downloaded from Nanostring website. Then we need the count files. We have the expression mat files. We need the mat file, and also we need the field view file. In this file, it contains the positions for the 30 images. So let's load the non-5 repeater 1 data. If you download the non-5 repeater 2 data, you can use the code I set here to load the data and create a data object. So now we use the non-5 repeater 1 dataset and created a data object. We can have a look at the A data object. You can see we have 100,292 cells, and we have 980 genes. So this is the original data. We haven't performed any quantity control analysis yet. So now we can perform the quantity control analysis. Because the in situ hybridization also designed the negative probers as the control probers. So we can get the negative probers information from the data. Then we can use the calculator QC matrix to calculate a matrix for quality control analysis. So once we got all the quality control metrics, we can calculate the percentage of unassigned negative probes transcripts. So later we can use this information to estimate the false discovery rate. So you can see it is around 22% for the negative probes total count. So now we can use the quality control metric to filter out the low quality cells. First, we can make some histoplot to show the total count per cell and the unique transcripts per cell, and also the transcripts per field of view. So let's run this code first, then we can explain the data using the histoplot. So you can see we generated 3 histoplots. The first one is total transcripts per cell. The x-axis is the total count per cell. You can see the highest count per cell is around 2,500. The y-axis shows the cell numbers that have the same count. For example, you can see for the cells that have 500 total count, it is around 800 cells. The second histoplot shows the unique gene count per cell. You can see the genes that expressed in different cells. Their cells has 0 count, and the highest count is 500. So here is the total number of cells that have the same number of genes detected in this analysis. You can see majority of cells have the gene expression around the 100. So the last histoplot to show the total count for each field of view. You can see in total we have 30 field of view. You can see we have 4 field of view have similar number of count in this area, then 7 field of view have similar count number in this area. So from 3 figures, I hope you understand the geometrics data. So this is the histoplot. In single-cell RNA sequencing data analysis, we also use the variant plot. We can also plot the genes by count and the total count for the variant plot. Let's run this code. You can see we generated the variant plot to show the cells that express the gene numbers. You can see the highest number is around 500. Majority of cells have gene expression around 100. Then here is the total count. The highest count is 2,500. So this is a different image to show the genes by counts and the total counts. So from both histoplot and the variant plot, we knew the distribution of the quality control matrix. So now we can set the threshold to filter the data. According to the online tutorial, we normally set the minimum count as 100. This is the total count for each cell. Then we set the minimum cells that express a gene as 500. The threshold you set here will affect the cell type's clustering, but if you don't perform the filterization, you can still see all the cells and the spatial data. You can change the threshold for your data analysis. So let's use the minimum count as 100 and the minimum cells for gene expression as 500 to filter the data. Let's run. After the filtering, we can have a look at the data object now. You can see now we only have 83,037 cells, but we still have the same number of genes. So even we set the minimum cells number as 500 for the gene expression, we didn't filter out any gene during the filterization process. So we performed the quality control. In next step, we can run a standard Squidpy workflow. Let's run it first. You can see for the standard Squidpy workflow, first we normalize the data, then logarithmize the data. In single-cell RNA sequencing data analysis, because we have lots of genes, then you can perform this step to identify the highly variable genes. But for the Nanostring dataset, we only have 980 genes, so we will skip this step. If we skip this step, we will use all the genes as highly variable features for next step PCA analysis. So after PCA, we find the neighbors, then run UMAP. Then we use the Laden method to cluster the cells into subgroups. So you can see we finished the standard Squidpy workflow analysis. Now we can plot the UMAP to see the cell clusters. Let's run. We can go down. You can see I used the resolution 0.3 in this analysis. Then we have 9 cell clusters in total. So in my previous video tutorial, I used this dataset to show you how to analyze spatial dataset using the thread package. So we know the cell types and the mark genes for this dataset. So we can use some mark genes to identify the cell types in each cell cluster. You can see first we can use the KRT17. It is the mark gene for the tumor cells. EPCAM is the mark gene for the epithelial cells, and the DCN is the mark gene for fibroblasts. Let's run those mark genes first. You can see we generated 3 figures. You can see cluster 0 are the tumor cells. Because the tumor cells were derived from the epithelial cells, you can see the tumor cells also express high level EPCAM. Then we can see a small cell cluster, cluster 7, as epithelial cells. Then cluster 3, you can see here, they express DCN, so they are fibroblasts in the lung. Then next we can run the mark genes for PCAM1. This is the mark gene for endothelial cells. RGS5, it is a mark gene for parasite. And the ACTA2 is a mark gene for smooth muscle cells. So the mark genes we list here will reveal cells in the blood vessels. Let's run. You can see this cell cluster. It is cell cluster 4. They express high-level PCAM1, RGS5, and ACTA2. So cells in cluster 4 are the cells that are associated with the blood vessels. So here are the mark genes for cells in the blood vessels. Next, we can plot the PTPRC CD52, CD79A. They are marked genes for immune cells. We can run. You can see the rest of cells. They express the marked genes for PTPRC, CD52, and the cells in these two clusters, they express CD79A, so they are B cells. So I use the different cell type mark genes to show you how to identify the cell types in the cell clusters. So for today's demonstration, I'm not going to show you how to annotate the cell clusters. If you want to annotate the cell clusters, you can watch my previous video tutorial for the Squidpy package. In my Squidpy video tutorial, I also showed you how to identify mark genes for each cell cluster using the findMarks function. That function is very helpful if you are not familiar with the mark genes for your dataset. So we analyze the gene expression data and cluster the cells into subgroups. We have 9 groups here. And I use the mark genes to show you the cell types in different cell clusters. So now we can use the spatial plot function to look at the cells and the cell types on the images. So first we can have a look at all the images using the immunohistochemistry data. Because in this dataset, they used 4 antibodies and performed the immunohistochemistry analysis. The immunostaining data can help separate the individual cells. Then we will generate more accurate data for the cell segmentation. As I showed you in the folder for the immunostaining, the best staining in this dataset is for the pan-cytokeratin antibody. We can use the staining result for the pan-cytokeratin antibody to visualize all the images. In total, we have 30 images in this dataset. We can run. Because we are going to plot 30 images, it will take time to run this step. Okay, you can see we have all the images. In total, we have 30 images here from the dataset. The images show the immunohistochemistry result for the cytokeratin expression, and the cytokeratin protein is largely expressed on the tumor cells and the epithelial cells. Because we plotted 30 images here, if you want, you can also plot the individual images. For example, we can use the cytokeratin result again, and we know the field view library ID. In total, we have number 1 to number 30. For example, here we can just use the library ID 12 and 16 to plot the images. For images 12 and 16, let's run. Okay, you can see now it is much quicker because we only plotted 2 images. So the first one shows cytokeratin expression on image 12, and the second image shows cytokeratin expression on image 16. This is just a demonstration. You can try to plot different images. If you want, you can also zoom in to crop the images. In this way, you need to set the crop coordinators. Then you can see the cells in more details in this area. We can use this area as the example for the demonstration. We can run. can see we zoomed in this area. Now we can see individual cells. We can see the original images for the immunostaining. And also we can see the cell boundaries for the cell segmentation analysis. As I said, we have 4 antibody staining. If you want, you can also plot images for other antibodies. We can use the CD45 as an example. Again, we are going to use the image 12 and 16. We can plot the antibody staining result for CD45. So you can see the images. Here are the staining results for CD45 on images 12 and image 16. If you want, you can plot other antibodies like CD3 and the membrane staining. Because we cluster the cell types, in my last demonstration, we can plot the cell types on the image. Here we need to set the color as the Nathan clusters because we performed the cell clustering using the Nathan method, and the cell cluster column was labeled as Nathan. So we can set the color argument as Nathan to plot the cell types on the image. Again, we are going to use image 12 and image 16 for demonstration. If you want to plot other images, you can change the library ID here for different images. Let's run the last plot for today's demonstration. So you can see now we generated images to show the cell clusters. You can see we have 9 cell clusters here on image 12 and image 16. Okay, I use the non-cancer dataset and show you how to analyze the Nanostring CosMx dataset using the Squidpy package. I showed you how to load the data and create a data object. We performed the quality control analysis, then clustered the cells into different groups. Then I use the spatial plot function to show you how to visualize the cell clusters and the spatial data. I'm going to stop here for this demonstration. You have analyzed the A dataset. If you want, you can read the online tutorial and use this analyzed A data object and perform more analysis, which will depend on your research questions. So please subscribe my channel if you like my videos, and also share my videos with your friends. Thank you.